And our next session, we're going to talk about one of their most important successes. It's called AlphaFold, and it is something, I'm not exaggerating here, that is, I think, one of the most important scientific breakthroughs in living memory. Now, admittedly, that depends on how old you are, but it's a very, very big deal. I mean, these guys, when they do conferences in front of scientists, they crowd surf their way out. Okay, that's sort of the level of people you're going to be talking to today. Right, so in a moment, you are going to hear from Anna Koivuniemi, who's head of Google DeepMind Impact Accelerator, which is using AI to address social and environmental challenges. And then we're going to be joined on the stage by two renowned scientists who are using AlphaFold to transform innovation in their respective fields.
Hundreds of millions of years. I'm going to talk about AlphaFold, but before that, I want to share with you concrete examples how machines learn. That's what Doyle tells is the basic of AI. There's been a lot of talk on football on the Zeitgeist, expressing our interest on the sport, and some of you may have been in the session of Thierry Henry or Laura George yesterday. Impressive talks. I'm going to share with you my favorite football play. Let's look at it. Looks fun. They might not be as good yet as Arsenal, but we're working on that. It looks very much fun, but there is actually some hard science going on here. We did not tell these robots to kick or to move right, like some of you have a warehouse automation robots that are programmed in that way. We actually gave them a goal, score, win the game, and we gave them some data to learn from. And they learned, they failed, they did it again, as we humans do, and they scored. And there was something even more amazing happening when we followed them. They came up with the moves they did not see before. Now, this is AI. This is one type of AI where we reward machines to do what we want them to do. And there is so many other types of AI, learning on patterns of fuzzy pictures and learning actually a lot of data to come up with the patterns. That's what helped us to crack the AlphaFold, the protein folding. Now, before I told about AlphaFold, what's the big fuss about the proteins? My teenage son thinks that they actually something to eat to build a muscle, but there's so much more. They actually biology machines. They're part of the whole actually life what we are. They make my heart tick. They make the blood flow. They give the oxygen. One of my colleagues actually said that humans would be a bag of water if we wouldn't have proteins. They actually help also plants and animals. They will make plants to digest the CO2, tell them when to grow a leaf, and then also tell them how to protect themselves against drought. Understanding how this protein works unlocks a lot of opportunities to live better. And that's why we started also the work on AlphaFold. But there was a big problem to start with. These proteins, which are amino acid chains, they come up to this very beautiful but complex 3D structure, which is very difficult to find out. And before AlphaFold, the researchers might have spent the whole PhD on it, 3 to 5 years for a single protein. Janet actually just told me, well, some of them spent 10 to 20 years and never cracked it. So it was not an easy problem to crack. And they were using expensive machines, which not everybody had access, and not every problem could use. There are actually over 200 million proteins in the world. So imagine if we would have done it traditional made, it would have taken hundreds of millions of years. Like these football playing robots, we didn't give AlphaFold a guidance how to crack the protein folding. We actually gave it 170,000 existing structures and said, find the pattern from the data. And it just did that. We were able to do this because there was data. Humans had cracked 170,000 proteins, which entities like EMBL, EBI had encouraged scientists to put to the protein data bank. So there was existing data the machine was able to use to crack the code. Without that data bank, there would have not been AlphaFold. And now there is. Today, AlphaFold has provided 200 million proteins, so all the proteins we currently know, to researchers to use them. And they can use it freely. They don't need any machine learning knowledge to use it, and they don't need compute to get access to it. Every quarter, my team is looking how these researchers are using AlphaFold. And it's a super exciting day, actually, for us, because we typically find things we never imagined it could have use for. They're using it to look at the reasons for malaria, to accelerate the malaria drug development. They are accelerating cancer drug development. They're looking at how crops like rice can protect themselves better when they found the protein structure. Here's the one very recent example, which is one of my favorite. When I was 10, I'm from north of Finland, so I was trying to grow tomatoes in our summer house. Now, you can imagine growing tomatoes in Finland is not really a perfect thing. And it was a summer of a heartbreak. They tend to die on me. And we found this group of researchers in Oxford that were looking at defense mechanism tomatoes have against these diseases. And it turned out to be a very, very interesting problem because tomatoes, the tasty things, have very complex mechanism how they actually protect themselves in the diseases, driven by proteins. And the diseases are blocking these proteins to work. The PhDs that we talked to, you can imagine this dusty room with the whole books of tomatoes, were explaining that there is the difficulty is actually to find these proteins that are defending the tomatoes. And they said they actually work is a little bit shot in the dark, that the last new fundamental protein was found 2004, and it took 4 to 5 years to find it. Since then, there has been no new protein found to defend, which defends tomatoes, until now. These researchers use AlphaFold, and they were so excited about it, to scan 23,000 proteins within a few weeks, and thousands of proteins in a few weeks to find 23 that were driving the defense mechanism of the tomatoes. And they said they found 4 new ones. Remember, it was 20 years ago when the last new one was found. They were so excited, and they are now planning to expand that to other crops and other diseases for tomatoes. And you can assure that I left the meeting with the respect of tomatoes, and maybe one day, I will try to grow them again. This is kind of things that get super excited when we see that this technology is used in real life to advance science. But it isn't everything. I'm super proud to share that last week we announced a new version of AlphaFold. And what does it do is actually not only explain the protein structure, but also how these proteins interact in this biology machine with the molecules like RNA, DNA, and small molecules. And that gives an additional information to the scientists of the functionality of proteins and hopefully advance the research further. You bet I cannot wait the next quarter when we look where it's used. AlphaFold is, for me, a great example how AI can benefit humanity. It's taken what humans has done, what humans have scanned the proteins, and made unbelievable progress within few years. That covers hundreds of millions of years. Imagine if we could use AI to solve some of the big issues we have the society in now, to benefit the whole humanity. Thank you.
Thank you very much, Anna. Now, we are going to be joined on the stage by a couple of world-renowned scientists to talk through this a little bit more. But I wanted to ask you, actually, while they're just getting the chairs ready, how do you make sure that this technology is in the hands of the people who can use it, the scientists and the researchers? Is it sort of a build and they will come situation?
It's a brilliant question. And to be honest, my scientists sometimes think that you release a paper and everybody knows how to use it. We have built an interface together with the EMBL, and now we have launched a server where we looked at how the researchers are using the AlphaFold and made it super easy for them to use. So they don't need to read AI code. It's just actually a visual picture they can turn and even integrate to their work structures. And Janet just told me that how important that we translate the machine learning uncertainty of the answer to colors, so they can see where the answer is certain and where less certain. But the part of the question also that this works with the people who use the traditional expensive machines to scan the protein, but there are a lot of problems and people in the world, especially in the global south, who never used protein folding before. So we have launched now a training model and are looking with these stories like tomatoes, encouraging scientists to look where you can actually use AlphaFold, because you can use it in such a wide variety of the science topics.
Absolutely. Well, hopefully we'll hear more about that. If you want to take a seat. I need to welcome onto the stage our two other panellists. If you want to take a seat. Yeah. We have Professor John McGeehan, who tackles some of the most pressing environmental challenges of our time using biology. He was a director for the Centre of Enzyme Innovation at the University of Portsmouth, where they are hunting for enzymes that can eat plastic as a way to tackle plastic pollution. We are also joined by Dame Janet Thornton, who is a world-renowned scientist, a pioneer in the medical sciences, celebrated for her groundbreaking research in protein structure. She's also, in case the damehood didn't give it away, a very big deal, okay? And no one really is better placed than her to explain the potential of AlphaFold to drug discovery or to understand diseases. So please join me in welcoming them both to the stage. Janet, I think I will start with you, if I may. If you could just summarise for us, why are protein structures so important? Just a simple one to begin with.
They do everything in life, basically. No organism, you or the trees outside, can function without proteins. Proteins allow us to smell, to hear, to see, everything. They provide our defence, and they're the targets for most drugs when things go wrong, to put things right. So proteins, and when you look at a person, actually, what you see is predominantly protein. And so these wonderful, we've lost our pictures of proteins, unfortunately, but they are wonderful structures.
Oh,
Here we are.
Look at that.
We have a structure.
Dame Janet says it, and it happens immediately. Yes. So before the days of AlphaFold, then, what was it like trying to discover the shape of a protein?
So when I started many years ago, there were only 20 protein structures known in the whole world. 20 structures. And now we have 200,000 experimentally determined structures. And of course, with AlphaFold, we have over 2 million structures, 200 million. So the whole world has changed. And I often think it's a little bit like, you all know about the James Webb Telescope and how it allows us to see into the far distance of the universe. Well, AlphaFold, in a way, allows us to see these structures in all their beautiful, complicated detail that allow them to produce life. And the fact that our proteins are almost identical, not the same, but very similar to those in the trees, in ants, in COVID, in all sorts of, every living organism is based on the same molecules, and the proteins are the things that really matter in my world. The DNA transmits the information from mother to child, father to child, but the proteins actually translate that into life.
A telescope to see the fundamental building blocks of life, almost.
It is.
Yeah.
It's wonderful.
So what does this mean then? What does AlphaFold mean? What will the impact be on science, but also on society?
So I think the impact on science already is on biology, on the life sciences, is huge, on research. So all my colleagues, they're all aware of AlphaFold. They all use it to look at every biologist has a special protein that they're interested in. And now they can look at these spectacular structures. And so they can understand how their protein works. And so in the structural biology world that we come from, this has changed things enormously. For society, for biology, not just... So I've always thought in terms of 3 dimensions because that's how proteins work. They have to be in 3D. But most biologists have thought about the sequence of the protein, the linear sequence, and not in 3 dimensions. But
like
of course,
if you unraveled this into a ribbon.
Exactly, exactly. And, you know, we can't understand, we are 3-dimensional objects, as you all know. You know, how come we managed to evolve 5 fingers, etc.? And so these, for the biologists now, with their favorite protein, they will go and look in the AlphaFold database at EBI and look at their protein and look at what it looks like in 3 dimensions. So that's a huge difference. In terms of society, I think that impact has really yet to be felt. But my goodness me, it will be. Because the impact on understanding the basics of biology is enormous. And from that will come a much better understanding of diseases and hopefully the cures, etc., that will come from that. So the impact is yet, I think, to be seen fully. But there is no... It's strange in science. Some things you can predict, and some things you can't. And this is one of those predictables. It will change medicine.
Absolutely.
Yeah.
But it will also change, I mean, other situations in which biology can have an impact, like enzymes, for example, John.
Indeed.
Can you tell us a little bit about what your work was like before AlphaFold came along and how AlphaFold changed things?
Yes, absolutely. I mean, as Janet has said, it's really been quite transformational for our branch of science. So I think it was David Attenborough who really put plastic pollution into the public persona, and now we understand the amount of plastic that's in the world, and we really need to mitigate that through technological solutions as well as behaviour change. But there was a discovery a few years back, quite a remarkable discovery in Japan by a group, Yoshida and his colleagues, who were looking for bacteria that were growing in a plastic recycling dump near Osaka. And what they discovered is a bacterium that was living in that dump, not off the sugary bits of the leftover fizzy drinks, but it was actually eating the plastic bottles themselves. So it was using these PET plastic bottles as a food and an energy source, which was really just a remarkable discovery. And this was because these bacteria were generating enzymes to break that material down. So to just understand a little bit about what that material is, plastic like PET is made of 2 molecules from fossil fuels, oil and gas, terephthalic acid and ethylene glycol. And they're linked together in long chains with an ester bond. This is a molecular bond that holds them together. If you've got lots of ester bonds in a row, you've got a polyester. So if you look at your jacket or your blouse, that's what a polyester is. A polyester is the same material as this carpet, these textiles, and the ubiquitous polluting plastic bottle. And that's fascinating because in nature, there's polyester and ester bonds out there. The surface of plant leaves is covered in a natural polyester. So nature is an incredible recycler. It wastes nothing, and everything is recycled, albeit at a slower pace than we're used to. And we've kind of upset that system by creating material that lasts for hundreds of years. But these enzymes have the ability to break down that plastic, and the idea is that by understanding their 3-dimensional structure, you can make them faster. So the enzymes they found in that plastic recycling dump do break down plastic, but not at an industrial scale, not fast enough. So what we're able to do then is, using that molecular blueprint, that 3D structure, is actually understand the shape. That allows us to make a faster enzyme. And it also allows us to go and search for other enzymes out in nature. And we found 100 of these. And as was said before, it takes about 6 months or a year to get one of these structures. So when DeepMind came along and won that competition for protein folding, the first thing I did was email Demis and said, Demis, we've got 100 enzymes, we've got a big problem, can you help us? Not expecting an answer, he's a very busy person. And he came straight back and said, John, speak to the AlphaFold team, John Jumper and Catherine and Agatha, and see if they can help. So they said, yes, John, this looks like a great problem. So on Friday, I sent 100 sequences to DeepMind. And then on Monday morning, I came into the office and bing, this email from DeepMind, 100 structures arrived in my inbox. And this literally has taken us 10, 20 years advance in one day. And it turned out that the structures that we were taking about 6 months to make, they could now do in 2 minutes. Completely transformational. And that we now are generating enzymes for all different types of plastics for recycling.
Paint me a picture then here. What's the sort of the vision of the future that you have in your mind, of the wider implications of this?
So this is one AlphaFold story, right? Plastic-eating enzymes. But nature, these enzymes are, nature is exquisitely specific. We heard, about COVID yesterday and how an antibody can recognize a particular virus molecule. So it's a bit like, it's often described as a lock and key. So we have an enzyme or a protein that recognizes one bit of the whole genome and switches on a gene at the right place. We have enzymes that exquisitely bind to only specific molecules and drugs. So what AlphaFold has given us is instant access to all those 3D structures of the lock. Last week, AlphaFold 3 came out, which arguably now gives us the three-dimensional shape of the keys. And having both of those parts of information I can't even describe. It was described yesterday as exciting, and I would describe this as completely transformational. As Janet said, I don't think we quite know how far this is going to take us, but it's massively accelerating science. I can't think of a more exciting time for AI to accelerate research.
I think that's it, because I think often it's very easy to get bogged down in the stuff that's going on, but actually there are real reasons to be optimistic about the future. And the implications of this are very far-reaching, Anna.
Yes.
What are you hoping to see? Paint me your picture of the future.
Like I said, every quarter I get surprised and I get enthusiastic about what the researchers are using that. I think there will be, for me, 3 things. First of all, that we continue advancing and accelerating the research and the ideas we have been discussing about in order to get real-life impact. So I think it would be fantastic to share a story that really happened. But the research is long and it takes years, and I hope that still in coming years we would see something concrete, and I do have my hopes very high, not only on you. The second thing what I would hope is that we see the people who never use, like I said, there's a lot of researchers who never heard about, you say they're looking at the string, not the structure, so that we can excite those researchers to use the tool or use basically the information they never had before and do really breakthrough innovation we could have never done before. So that would be my second wish. And a third wish, and we discussed this also, is that in order to have a people who can use and know this is available, we need to actually make sure that every scientist knows a bit about AI. So I would hope that in our education system and when we talk to scientists, we can also sparkle the interest and provide the content them to learn about to use this tool.
And Janet, looking forward, looking ahead, are there other problems that you're hopeful that you'll see AI or AlphaFold try and tackle? If you've got a shopping list, what else do you want on the shopping list?
So, 2 or 3 things. First of all,
be greedy. Go on.
Yeah, I will be very greedy. I think when we go to see our GP, they will be using AI-powered tools and using the knowledge gained from these structures to actually improve their diagnosis and prognosis, and hopefully to be able to give us medication that will solve the problem. So the whole of the sort of medical world, I think, will be revolutionized. Even in my lifetime, I think that's going to happen. I think the other is the agricultural world, the biodiversity world. So one of the great things is that we can now measure measured biodiversity using molecular technologies. And so we can predict the impact of building a railway or building a new thing, a new factory or something. And so using these tools to learn, we can really change the way we look at ecology and look at producing crops. You talked about the tomatoes, but this applies to everything we eat, all the plants. We heard about the science of healthy eating yesterday, and eating the proteins in the plants. And, you know, that's really important. And I think the AI approaches to improving design, both of the specific enzymes for plastics, but many, many other things for clearing up waste everywhere, and etc. It's phenomenal. So I think it really, this combination of biology is made, or AI is made for biology. I don't know which way round it is, because it's because of evolution, and it's very data rich, and traditionally we have shared our data in biology. Now that might change because the commercial gains could be huge from this, but I think having open data, and I would like to commend DeepMind and Google for making these structures available to everybody. And that has had a huge impact, and it's where the medical field needs to progress. We need to share medical data while keeping it confidential so that we can progress and really understand these diseases.
Absolutely. Lots of really exciting stuff on the horizon. Thank you very much to all of you, Anna, Janet, and John. Thank you very much indeed. Amazing stuff.